years now, and we know the 40 most common mutations in the populations in the US. So one of these tests on the DNA of the patient, if you have a patient for which the symptoms tell you that it's likely to be cystic fibrosis, then you test these 43 mutations and it costs you $3,000. And if you don't find any mutation, then you have to have some other idea, do some other tests. And so in some cases, you end up spending a lot of time doing test after test and not having any diagnosis. So given the current power of new generation sequencing, it's of course tempting to try to do all the tests or a number of tests immediately. And I think I have examples in the next slide. So actually what you can do, you can either have specialized panels, so a test that looks at all genes possibly involved in severe combined immune deficiency, or all genes possibly involved in heart problems, or all genes possibly involved in deafness, or many other cases. Or you can actually try to look at all, almost all known Mendelian diseases. So that I think I have examples of these two approaches. So this is again offered by a company. It's called Children's Mercy Center, but it is a commercial company. And so here you have the cases, what they say about the cases in which this kind of test is likely to be useful, basically where there is evidence that it's a genetic disease. Many tests have already been done without conclusive results, and suspected diagnosis would change treatment, of course. And I think the next slide shows one of their panels. So this is their so-called comprehensive SCID panel for immune deficiency. So they've assembled have 18 genes known to be involved in some immune deficiencies. And so if you send the DNA, they will do the tests they will sequence all these 80 genes and look for mutation, and it will cost almost $4,000, but again, it may be quite worth it. And again, this is only possible because you can do a lot of sequence without spending too much time and too much money. Next slide. So in this case, that company, the name, as you see, is Good Start Genetics, and they advertise the similar tests for a large number of mutations as carrier screening, which means prospective parents before they conceive a child check that they are not both carriers of mutation in the same gene. So I'm not sure what I have after that, but this approach has been used in one specific case within the Ashkenazi population. Maybe we will come to it. So this is another possibility. Next slide. And this is maybe a more advanced test. This is again by Children's Mercy Hospital, and as you see here, they've been able to set up a test that looks for genes involved in almost 600 rare childhood diseases. Again, these are all monogenic diseases where there's a single gene involved, and this gene is known. And I think I have one example of using this test. It was actually not used as a carrier screening before having a child. It was used to diagnose children with a severe problem. So again, this is the case. You have two children with severe ataxia problems, uncontrolled movements, and these kids had already been through several years of DNA testing, spending a lot of money, and not finding mutations in any of the 4 or 5 genes that had been tested. And so using the test that was shown in the preceding slide, a mutation was found in a gene that is known to be involved in movement control. And it turned out then by sequencing, running the test of the parents, that both parents were carriers. So they were both heterozygous. They had 1 copy of the mutated gene, 1 copy of the normal gene, so they were normal, but the child had inherited, the 2 children had inherited the 2 non-functional versions of the gene. And it turns out that in that case, knowledge of the gene involved suggested a treatment with a drug that was effective. So again, knowledge in that case led to more effective treatment. Okay, so as the examples I've given show, this is a way to do pediatrics diagnostics to avoid so-called diagnostic odysseys, which lasts 4 years, test after test, and no gene found and no treatment can be effective. Can be used for carrier testing for future parents. And some people are talking about extending it to systematic screening of embryos in vitro fertilization. You could imagine that you would test many genes in embryos produced by in vitro fertilization before implanting one of these embryos. It is already possible to obtain a complete DNA sequence from a single cell, so it is possible to do that. Whether one should do it is another question. Okay, next slide. So, of course, in all this one must, there are a number of issues about the quality of the sequence, whether you can really trust the sequence when you find a mutation, you really test it with the gold standard method, which is a lot slower and more expensive. There's a trade-off between quality and cost, but I will discuss this in detail. There's also a lot of work to be done on the interpretation. You cannot spend months of informatician time to understand each case. You have to have good programs that produce results that are clearly understandable. In the clinic, the time factor is often critical. You cannot wait 3 months for the result. You need, if you are using this for to guide treatment, you need things to be done in a regulated environment. In the USA, it's the so-called CLIA environment, and then you need to handle the cost and the reimbursement of these tests by insurance companies or by the national health system. So actually, this is really being worked out. The next slide shows a few news items showing hospitals or centers that have started using new generation sequencing in actual clinical practice, and that are working out the details, the procedures, the administrative handling to really put it into full practice. Okay, finally we come to the ethical issues. Yes. So probably the major new feature that comes up is so-called incidental findings, meaning you're looking for one thing and you find something else. And this has impact on informed consent, and it falls into the general issue of genetic privacy. So I'm going to discuss this in the last 10 minutes or so. Next slide, please. So this is, well, an example of incidental finding. You have a baby girl with signs of mental retardation. You have some reason to think that this is genetic, maybe because an older sibling was also mentally retarded. Anyway, you end up sequencing this girl's exome, And actually you find that she is carrying a mutated BRCA1 gene, which means she's at high risk of breast cancer, which means that maybe among her sisters or extended family, there are also people who are carrying this mutated gene and who don't know it, but who should know it because it's important for their healthcare. So you found something you didn't expect. What do you do with it? Whom do you tell? Who has the right to know? Who has the right not to know? So of course, as I indicated in the next slide, this is not a completely new problem, because even in conventional testing, you sometimes find things that you were not looking for. But with new generation sequencing, you get easily 1,000 times more data than usually. So that means that incidental findings are going to be present all the time. Every time you do one of these diagnoses, you may find what you're looking for, but you're always going to find things that you were not specifically looking for, and you will have to deal with that, to know what to do with that. And that means that the people whose DNA you sequence should know about that beforehand. When they agree that you're going to sequence their DNA, they should know that unexpected things will be found, and they must have decided beforehand what they want you to do with it. So next time, just extend a little the question. So will the incidental findings be reported? Then which incidental findings do you report? Do you report only things that you are sure have very serious health consequences, or do you report things which are a little more shaky, or do you report more or less everything? And so how much counseling do you need to make the patients understand what's involved before they sign the consent form? Well, how long is the consent form? Do you end up with a 90-page consent form? And how much counseling do you do once you get those unexpected results to explain what they mean and what they don't mean to the patients? It's also an important question because the DNA, you sequence DNA, except for a cancer patient, you sequence a child's DNA. This is permanent information. This sequence is not going to change. But 1 year from now, new knowledge will have been obtained. And so some things in this sequence that did not tell you anything today, a year from now may tell you something. So should you reinterpret the sequence every year or every 2 years, and what should you do with the results you obtain? And again, the patients should certainly be aware of that. So it's not a completely new problem, but its magnitude is much higher with this technology. In preparing this talk, I found a couple of recent papers that I thought are quite interesting. Both have someone called Tabor as the first author, were published in American Journal of Medical Genetics in 2001, and there's another one in 2012 about these issues. And the second, this paper is more general, and the second paper is more experimental about the consent form. And I think they list a number of questions. When you look at existing consent forms and wonder if they are adequate for diagnostics involving NGS. And also, next slide, this is another table from the same paper that indicates a number of necessary areas of research as we are getting this technology into clinical practice. And I think these two papers are pretty, I don't know if you had a chance to look at them, but they give a pretty good picture. Okay, so of course, a number of people, these institutions that have started using new generation sequencing in the clinic have come up against these problems, and so a number of institutions are working on these problems. There is quite an interesting survey being done by Sanger Institute. I will show something about it. There is a specific project in Germany. That's work from the Presidential Bioethics Panel in the US. There's also been a special call for proposals with almost $6 million funding from NHGRI for research on these topics. So there's a fair amount of activity. Next slide shows actually the Sanger survey. It's actually a survey to the general public. So each and every one of us can access the Sanger website and answer this survey. And it's useful to do it. I intend to do it. As a patient, would you like to know about what pertinent findings? Presumably you want to know, because that's what the test is supposed to look for. But do you want to know about incidental findings? How do you categorize incidental findings? How do you understand risk? Do you want to be able to get the raw data and maybe give it to somebody else for interpretation? What do you consider the duty of the genomic researchers? And so on and so forth. Do you consent to genomic research being done with your sequence? And so on. So there will be, I think there will be quite a lot of also ELSI activities around this topic. Next slide, please. Okay, just maybe a fun slide, just to emphasize that DNA sequence information is really sensitive information, sensitive in the sense of it reveals a lot of things. This is something that you may have heard about when Obama was running against Hillary Clinton in the Democratic primaries in 2008. Someone managed to get hold of a half-eaten waffle from Obama's breakfast in some restaurant, and he or she, I don't know, put it on sale on eBay, and he put it on sale on eBay saying this waffle contains Obama's DNA, and so of course the market value of this half-eaten waffle was that you could access, in principle, Obama's DNA, maybe find out if, in the mind of people in the US, if he's genetically a Muslim. This was a big idea, or maybe if he has, more seriously, if he's the carrier of some serious disease that might incapacitate him as President of the United States. But just to indicate that in reality, and also in more in people's minds, the DNA sequences contains a lot of information. And this is just two examples of tests that are indeed offered to the public currently in the US and in a number of other countries. So-called secret paternity testing, where you don't have to tell anybody, you just send them a used toothbrush, find out if the person who has used the toothbrush is the father of a certain child. And also prenatal paternity testing, where you can actually, from a sample of a pregnant woman's birth, do a paternity testing on fetal DNA, which ends up in the mother's blood, and so find out if, let's say, you, the husband, are really the father of this child that is growing in your wife. So you can imagine the serious ethical problems this raises, but this is actually legal and is being done. So DNA is not just any body part. It is really information, and information is something important. Next slide, please. So this comes to the issue of genetic privacy. Also, once your DNA has been sequenced, you would like this sequence to be really confidential and to be looked at only by the people you decide have a right to look at it. But in practice, this is probably quite difficult to safeguard, because, as we all know from emails and WikiLeaks and this and that, files get copied very easily, informatic systems get hacked into. And so some people say that confidentiality cannot be guaranteed. And again, if we think this is the case, then the patients should know that before they agree to have their DNA sequenced. Okay, and I think coming to the last slide, or almost the last, okay. All the insurance issues, of course, will the insurance pay for the test? Will they get the information? Will they use it? Will they end up denying coverage, as has happened in the past? We spent a whole seminar talking about that. Next slide. Here's another cartoon. So you have DNA, a roller coaster ride for science going very fast, and bioethics trying to keep up. Next slide. Okay, that's the summary slide. So this is really a revolutionary technology because it's only a quantitative improvement, but the quantitative improvement is so huge that it really changes the nature of things. It's one case of quantity, quantitative changes producing qualitative changes. And the applications are really beginning. There's still a lot of choices to be made, many issues, new ethical problems. Which we all have to work on. And I think my last slide is just thank you and tell you that we live and work in a very beautiful place. You should visit us. This is the view from my new flat in Marseille. Wow. The swimming pool is not mine. I prefer to swim in this works.
Okay, thank you, Professor Jordan, for your very informative, intriguing, and stimulating talk. So we are now open the floor to everybody for discussion and question.
Yes.
Do you like your DNA be sequenced?
I would say I wouldn't mind, but I don't feel a great urge to have it done. But maybe if I was 25, I would feel differently. I feel that okay. I'm in the last third or quarter of my life, and I have had my children and my grandchildren, and I've lived my life.
But I think your sequence should be your genome should be sequenced because you have we have known what all you've done in your life, so we can have good search from the genome, right? And find some association. So I think we should be rather sequence for the people who are going to retire or go to the heaven, so that we can have a retrospective, post-cross-examination with their past behaviors or events.
I mean, if I was approached for being sequenced in a project that I think is scientifically interesting, I don't think I would mind. But as I said, I don't have I have a number of problems behind me rather than in front.
Yeah, sure.
So would you make your genome sequence available to the public? Also, the second follow-up question, in the case in France, would that be legally acceptable to make your genome sequence available to the public?
Okay. I would hesitate to make my DNA sequence available because of my children. I mean, I would have to check that they agree to it. I think currently in France it would not be legal because, well, normally in France at this time sequencing would come under the handling of genetic tests, and this can only be done for medical reasons or for in the framework of criminal or civil investigation. So I don't have the right to send my DNA to a lab for sequencing. This has a 3-year prison penalty and 750,000 euros, although it's never been done before. That's special.
Further questions? Okay, please.
You brought up the big problem of incidental findings. And you mentioned that it comes up anyway in practice without DNA sequencing. My question is the way doctors have dealt with incidental findings in the past, will they be able to apply that to genetic problems? Or is it just so much more information and new information that we'll need new sorts of guidelines about how to deal with incidental findings?
I don't really have extensive, I haven't seen an extensive study of how people have dealt in the past with incidental findings, but my understanding is that this is something that has come up from time to time and that people have dealt with more or less on an ad hoc basis. In some cases saying nothing, in some cases saying everything, depending on the MD and his relationship with the patient and the personality of the patient. And okay, the point that's made by a number of people, which I wanted to make here, is that this is going to be now very common, so there has to be some systematic way of dealing it. Otherwise, there will be lots of unevenness and deviations. But no, I don't know if this has been studied seriously by anybody, how it's been done in the past. I suspect it's also not being recorded generally.
I'm curious and interested about the Ashkenazi panel. So the Ashkenazi panel for DNA diagnostic.
Yes.
Okay. So I'm aware of the few genetic disease that is more common in Ashkenazi Jews, for example, some of the lysosomal storage disease and also the ELSI mutation. The reason I'm curious is that I'm aware that there are certain genetic diseases that are more common in certain ethnic groups in Taiwan, for example, Kejianen in Hakka. They have a higher incidence of G6PD deficiency and some of the blood disease, for example. I just don't see how this could be done in Taiwan or in specific group that because people always worry about stigmata, that tax associated with a certain group of genetic disease. So I'm just curious how these Ashkenazi panel come about and become commercially available and acceptable.
Well, the Ashkenazi case is a very special and interesting case. So the issue is a disease called Tay-Sachs disease, which is a horrible neurological disease where the kids die at age 1 or 2 after a life of suffering, short life of suffering. And this is a genetic disease, recessive disease, so it manifests itself if both parents are carers, in average 1 child out of 4. And it's actually highly prevalent, it was highly prevalent in the Jewish Ashkenazi population, probably because it's a recent mutation